Thank you for that introduction. It's most of my presentation, so. And it's Bob Levis. It's pronounced Levis. Happy to be here. James Brown and I met at a similar conference at Penn Medicine a few months ago, and he suggested I come out here, tell my story, and he said, come on out for the whole thing. And so I did, and I try to attend these events. I don't understand half of what's going on or what's being presented, but as a cancer patient, you know, smart patients get smart care, so you try to understand as much as you can, and you're always looking for what's coming down the pike, because as a cancer patient, you may be on one drug, may not last forever, so you want to know what might be next and have a plan. You can't always rely on, or just say, you know, okay, doctor, tell me what to do. You have to be prepared to make a decision. So again, smart patients get smart care. So I try to learn as much as I can about my disease and try to help others who are dealing with chronic lymphocytic leukemia. That's way ahead. I want to thank all of you who are in, you know, microbiology, biology, in genetic research, cellular research, for what you do. I would not be here otherwise. I would have died in 2013, probably, because my CLL had advanced so rapidly. But I've got 3 slides. Let me tell you my story. I was diagnosed in 2002. I was on a business assignment with a company called Air Products, running our largest division in Asia. I had the dream job. I was 51 years old, and you know, good time in life, personally, professionally. And the company had an annual physical that you were required to go through, blood work and so forth, which I always did. And in 2002, the blood work came back, and it was high white blood count. Okay, what's that mean? Everything else is okay. I feel fine. Nothing else is going on. But they said, you know, check it out. We need another blood sample. And they looked deeper and said, you could have a leukemia. And it's like, what? I was thinking maybe high cholesterol or something, but leukemia. So when I got that result, I was right there at Singapore University Hospital, and they said, we need to do a bone marrow, and we're going to bring me back the next day, and their procedure was to put you out for all day. And I said, well, can't you do that straight up? And they said, yeah, and it hurts a bit. And my wife's sitting there and says, he's pretty good with pain. So, you know, it's one of these looks. So I did it. And right there, it hurts. And in 16 years with CLL as a survivor, I've had about 15 bone marrows, and all of them straight up, just get it done. But it did show that I had leukemia, chronic lymphocytic leukemia. And right there, you say, well, geez, how long do I have to live? That's your first leukemia, you know, geez. And it was a real, you know, just knocks you back. You can't believe it. And then you start to do your homework. You start to do the research. You start to dig in, start to understand your disease. And I learned right up front that I've got the good leukemia. Good leukemia, 13q deletion, a slow progression, immunoglobulin, heavy chain, mutated. So those are all good things in a chronic lymphocytic leukemia because it's slow progressing. And it was. I remained in Singapore doing my job for the next 6 years and felt fine over the time. I started to get a few lymph nodes growing in you know, armpits and you know, in my clavicle, groin, but I was fine. But ultimately, by 2008, as you see, it had grown to the point where my white blood count, which is normally 12 to 18,000 was over 300,000. And what happens? It starts to crowd out your bone marrow. The other things you need that the bone marrow produces, hemoglobin, platelets, starts to crowd that out, and they start to fall off, and you can't live very long without those. So I started treatment with the then best available proven treatment through trials at MD Anderson using chemo and monoclonal antibodies, the fludarabine, cytoxan, rituxan, which worked. It was roughly 3 months of that, 4 months of that, and I got a complete remission. And felt good, went back to work, actually for a bioreactor generator company running their business in China, ABAC. But then in 2011, to my shock, I relapsed. It came back, and it came back with a vengeance. It had flipped to the most aggressive form of CLL, and a bone marrow biopsy showed that I had this TP53 deletion, which you find in many cancers. If you lose that gene, it's a tumor suppressor gene, and it allows a cancer to grow more rapidly, in simple terms. And it did grow more rapidly. By 2012, I needed to get some other treatment. I'll go back to 2011. That was the year that in the New York Times, there was an article about Carl June's work at Penn Medicine, and it said 3 CLL patients cured with their own killer T cells. And I said, well, that's timely. I need to contact Carl June. So I contacted Penn Medicine. I got his email. I sent him an email, gave him some background, and he sent me an email the next day. He said, you've only been treated once. You need to be treated again to even qualify. You need to fail that in order to get into one of our trials, and that's assuming we're going to open another trial. But he recommended that if I have another treatment, have it at Penn Medicine, get registered there, which I did, and I entered this trial in 2012, which was a randomized trial comparing ofatumumab with the drug that I actually wanted to be on, ibrutinib, which is a kinase inhibitor, which is an oral pill you take. It blocks a pathway for the formation of cancerous lymphocytes called the BTK pathway. After 8 weeks, I got randomized onto ofatumumab and didn't get on ibrutinib. After 8 weeks on ofatumumab, it didn't work. I was hurting in a bad way. The CLL was becoming very aggressive. I had a packed marrow. The Pharmacyclics, the company, was not crossing people over to this drug that was working wonderfully on other patients. I'd meet other patients who are on ibrutinib and say, how you doing? It's saving my life. How you doing? I'm dying here. But I couldn't get on ibrutinib. As you see, by 2013, rapid CLL progression, transfusion dependent for hemoglobin and platelets, multiple lymph nodes, and my marrow was packed, and my TP53 deletion was up to 72%. So talk about being at the right place at the right time. My CAR T therapy experiences, in March 12th, 2013, after giving up my T cells through a process called apheresis. They take your T cells from me. They have 2 probes in your arm. They take the whites out, T cells out, distill them, put the reds back in the other arm, and they go off to the lab. At that time, it was 8 weeks in the lab so that they could take my T cells, modify them with a viral vector, and bring them back, a vector that would be attached to my T cell that once they reinfused them back in me would go directly to a protein on the surface of cancerous lymphocytes called CD19, and it would kill it. That was the theory, and that's what I volunteered for as patient number 4. There were those first 3. In 2011, I was patient number 4. So I volunteered. I said, hey, what else am I going to do here? And I went through the whole trial. I had the response they described to me. They said, you're going to get very sick once these T cells start to kill. I had a lot, about 3 or 4 pounds of tumor burden in my lymph nodes. And they said, as that killing takes effect, you're going to get sick, very sick. You're going to get fevers. It's a cytokine response, and we're going to need to hospitalize you, which is exactly what happened. A week after I got my infusion, I was hospitalized in the hospital. My wife was there documenting some of this stuff. I was near comatose at times, 101 to 104. My heart rate was 100 beats per minute, 24/7 for 8 days. I lost about 18 pounds, couldn't eat, and the nurses who had not been trained about CAR T were thinking, this guy's dying here. We got to get x-rays. We got to take samples. And I'm saying, would you please read the trial document here? This is supposed to happen. Bring it on. Stop the Tylenol. You're giving me the sweats and the chills. And it was horrible. And my wife told me several things, that, I said or didn't, don't remember, through that whole experience. But by day 9, the fever broke, and my lymph nodes were back to normal. This happened in 9 days. And my platelets, which had been about 30,000, jumped to over 100,000. And my hemoglobin started to come back. I could breathe again. I had lost, all this weight, but what we knew had happened is that these T cells actually got inside, and not just my peripheral blood that's flowing through me and killed the CLL, it got into all my lymph nodes, killed them, but it got inside the marrow, which it's tough to get in the marrow, and it cleaned it out. A bone marrow biopsy, another one, a month later, showed that I was in complete remission. And it took me a couple months to get all the, you lose your, that much weight, you lose your, muscle strength. It took a couple months just to feel right again, but I was clean, complete remission. So it was celebration time. And we all took my family to Hawaii that Christmas. But didn't last forever. That's the nature of CLL. This is FDA approved for ALL, for non-Hodgkin's lymphoma. They're getting 80% plus complete remission rates. And in CLL, they're only getting about 26%. So I'm still an experimental patient. I relapsed in 2016. I went on finally that kinase inhibitor, ibrutinib, which stopped progression. It's very slow progressing, no lymph nodes. And then I got stable and decided to do CAR T again because they had another trial. They were looking for candidates who were on ibrutinib as the cytoreductive, sort of a cytoreductive conditioning prior to doing T-cell therapy again. And it was a different vector, different CAR T product, all at Penn Medicine. So I went through that thinking, give me the complete remission again. This worked once. But it didn't work completely. I had 40% CLL in my bone marrow going into the trial, and I had less than 5% after. And I'm still on ibrutinib. I'm stable on ibrutinib. And that should work for a while, but it doesn't work forever. There are other drugs, kinase inhibitors that block BCL2 pathways, venetoclax. And I'm going to continue on this path while I watch to see what's coming next in CAR T, because I believe in it, I support it, I help funding for it, I help bring different people and organizations together because I believe in it. And I'm hopeful for the future that that's going to be the answer ultimately for anyone with CLL. I have one more slide, and it's just some questions, some observations that I have made based on reading that research paper that Penn put out. I've read it about 3 times, highlighted it everywhere, because I'm 2 of the data points in here of the 41 patients. And so, if you come to the conclusion here, they say, well, we can probably cherry pick 26% based on their genetics and get a complete remission from them. And my challenge to them is, no, we want 100%. So I looked through it and I came up with these first 4 bullets that are questions. You know, can you enhance CD27 plus CD and suppress actually CD45RO CD8 T cells in order to get move from a non-responder to a complete responder? Can you control these biomarker signatures in some way? Because these are the things that make the difference between getting a complete remission, a partial remission, or no response at all, NR. And so you can read them there, but I sent this back to the Penn team, and they said, yes, yes, maybe, and yes. But we need more money, we need a cell sorter, and that's where, you know, the title, complicated and expensive, as Larry Cooper from Zyopharm said on the first day. It is complicated, it is expensive. A cell sorter itself is $700,000. And if they were to dedicate that to CLL and to answer some of these questions, they'd have to set up a GMP framework. Personnel costs would be total about $2 million to dig into that problem. And they're going to do it, you know. They're going to focus, you know, on, they're already doing it, working on it one at a time. I'm going to help them raise money to get another cell sorter. The other bullet here, universal CAR T cells. That's another hope for certainly CLL patients, blood cancer patients, all patients. An off-the-shelf product where you can use a CRISPR technology to knock out an endogenous T cell receptor to enable transfer of allogeneic CAR T cells from a single donor, healthy donor, into many unrelated recipients. There are a lot of people, well, not a lot of people, there are some people working on that technology. And then this final example is somebody I know, lentivirus integration CLL case, a CAR transgene integrated into one TET2 allele during cellular manufacturing. The patient possessed a pre-existing mutation in his other TET2 allele, led to a remarkable clonal expansion of a single CAR T cell, resulting in complete and durable remission. This individual has been in remission for 7 years. He was one of the first 3. And they still can find CAR T cells in his peripheral blood. In my case, my first time, they lasted for about 3 years, and the second time, CAR T, they lasted for about a month. I had a very mild cytokine response. So I still give my blood, experimental labs, bone marrow, to Penn. They're studying it, and I'm happy to do so. And that's my story. And again, thank you all for what you do.
Well, you were not kidding about being a smart patient. Wow. And I think with you, I love that you gave feedback on their paper about you in the entire trial. I love that. Have you thought about this? Well, I can only imagine you taking such an advocate position for yourself, just benefiting thousands of others. So it wasn't selfish. It's definitely going to pay off for so many other people that will be benefiting from what you're going through and from you asking those questions. So thank you for sharing your story. We do have, again, 2 microphones on each side if anyone does have any questions. Would you give any advice other than to somebody maybe facing the same situation you are? How would somebody, because you are getting such a heavy diagnosis that all of a sudden you have to deal with this, how does one then basically get a backbone saying, okay, I'm going to be my advocate? How do they start? What do they do?
I talk with patients every week, you know, on this subject, blood cancer patients, and so I get referrals sometimes from Penn or from friends who are dealing with something. And with what's going on, I start with, hey, there's an answer, you know. It's genetics. Get to a specialist. You know, they're digging deep, and you need to dig deep inside yourself. And find out more about your disease and figure out, put together a game plan like you would in anything, whether it's a ball game or a business, you know, plan. Have a game plan that you know you can trust and rely on, and surround yourself with good people, including the doctors that you're dealing with.
Good advice. Does anyone have any specific questions for Bob?
I'll add one more thing. There's 20,000 new cases of CLL in the US every year. So there's a market. And my hope is that we're able to solve and answer some of the questions that I've posed up there. And a vision for the future with all the new technology coming down the stream. And I've got 2 sons. I've got 3 grandchildren, 1 more on the way. So none of them have been genetically tested, but I hope none of them have to deal with what I have. But it's a possibility, the genetics of it all. I hope the future vision is that this is, becomes like a flu shot, like an individualized flu shot that is developed and carried and can be without the cytokine response. And that's a vision. So for you students out there, let's get there. Thank you.
Thank you.